you now, Rebecca.
Awesome. Thank you.
You're welcome.
I'd like to start today just to give a little bit of an intro about me and who I am. So I graduated with a Bachelor's of Science in Biomedical Engineering from Georgia Tech. And like Ryan said, I'm currently a PhD student in Biomedical Engineering at Northwestern. So what that means is I'm doing research full time. So I know we hear research a lot in the media, especially now. So I have a good relationship with that. And the reason I wanted to put this together is because there seems to kind of be a disconnect sometimes between people who aren't so familiar with what research means and what's actually going on. So I have work experience at Johnson & Johnson and Becton Dickinson in research and design. So I've been kind of on the end of the design of new medical device products and new pharmaceuticals. So I'm hoping that I can give some perspective there. And with that, I also have some research experience in infection mechanisms and other cellular diseases. So I'd like to keep this presentation more interactive, like I said. So if at any time you have questions, please just jump in. So I'd like to start with one question that I see a lot is, where did this virus come from? So coronaviruses in general, when you hear about SARS and MERS, are derived from bats. So actually over 89% of this virus's genome of COVID-19 has similarities with that of Chinese bats. So what this means is that it was likely native to bats and it transferred into another animal host. And then somewhere in between there, what happened is the virus mutated and gained the capabilities to infect humans. So although when I describe it as this sort of linear process, it sounds kind of simple, but in actuality, there's a lot of very, very complex biology and protein mutations that are involved in this process. So if you look at this diagram here, there's very specific sequences that are involved. And basically what happens is you get this perfect storm where the virus all of a sudden randomly gains the capability to infect different hosts, in this case humans. So it could have been transferred from a bat to a pig or something like that, where in the dense animal market, like we saw at Wuhan, there's a lot of different species in close proximity, and that makes it more likely that this virus could mutate. So number one, how do you protect yourself? This is something we've been hearing a lot about, disinfecting surfaces. One thing to think about is make sure the surfaces are clean before you disinfect them. Avoid touching your face. Wash your hands often. Ensure your hand sanitizer is at least 60% alcohol. Another thing is to make sure that the hand sanitizer or the disinfectants that you're using aren't expired. That's one common error that people will make when they're using things like bleach, is it'll actually be expired and then it's not as effective in actually killing the viruses off these surfaces. In general, the virus doesn't survive on surfaces particularly long, especially soft surfaces. So disinfecting things, maybe when you come home from the grocery store, isn't entirely necessary. But if it gives you that extra peace of mind, then go ahead and do it to feel safer. The number one thing is to avoid contact with individuals that may have the virus. I'll mention this again later, but up to 40% of individuals that are actually infected have no symptoms at all. They're asymptomatic. And that's part of what makes this virus so much more transmittable than things like the flu, which commonly with the flu or other kinds of viruses, someone actually has to have symptoms in order to transmit it or spread it. So that comes into why we actually have all the shelter in place and social distancing going on, because of the dangers that people who are asymptomatic pose. I'd also like to talk about the cloth face coverings. There's been quite a few misconceptions I've seen about why we're wearing these and what the purpose is. Really, it's to target that 40% of infected people who don't demonstrate any symptoms. So the cloth face coverings actually trap the respiratory particles that might come out of your mouth or your nose during coughing or sneezing and just regular breathing, and they trap them in the mask, so they're less likely to transmit through the air or get on other surfaces when you go out to places like the grocery store and the essential places that we still need to go. So that's important to keep in mind, is that the cloth face coverings don't actually protect you. So when you go out, you still need to make sure that you're washing your hands really thoroughly and disinfecting surfaces to lower your own risk of getting infected. And with this in mind, don't wear the face coverings if you already have trouble breathing and this is not something that's physically feasible to you, or if you're not able to take the mask off without someone helping you. Also, children under 2 shouldn't be wearing the masks. And thinking about where we can get these, right now, the high duty N95s and healthcare masks should really be reserved for healthcare professionals. So this diagram here is interesting, where really anyone can create a mask even without sewing if you don't have any experience, just from an old t-shirt. Some things that you need to have when you're wearing these cloth face coverings: they should really be at least 1 layer of fabric. 2 is much better at trapping the respiratory particles. And then you can also fabricate the strings to tie around your head, and then there's no sewing involved. So this method I thought was really interesting because it'll stop you from having to necessarily take a trip out to a fabric store or a clothing store and buy the cloth to sew, especially if you don't have those resources handy. This is something that's really doable, as long as you have scissors and a shirt you're willing to sacrifice. Okay, so something else that I've gotten a lot of questions about is, how are people tested? How do the tests that we're performing actually detect if you have the virus or not? We've heard a lot about how these tests are hard to get, and on the news we've heard about different states that are doing better at testing than others. So the reason that testing is such a big topic right now is the reason patients are identified as having the virus is because it's a very advanced specific molecular biology technique that's being used to test whether people have the viruses. And it's called PCR, which stands for polymerase chain reaction. So this sounds like a lot, but the way this works, like I mentioned earlier, the genome of this virus is very similar to the genome of viruses that infect Chinese bats. So leveraging the fact that we understand this virus's genes and what its RNA and its DNA looks like, we take swabs from patients and then we try and look for strands or pieces of DNA that we know the virus has, and we're trying to identify that in a human patient. So the way this method works is we have to identify those pieces that we're looking for. In this diagram here, where the green would be the virus's whole DNA, these pink little blocks here and throughout the diagram are the specific sequence that we are looking for. So really what you're doing is trying to find Lego blocks that might fit onto this DNA, and then using that to identify how much virus DNA exists within a person, and then within that person, can you judge if they have the virus? So that's basically what PCR is doing, and the different testing kits all look at different pieces of DNA. They all use different Legos to try and determine whether a person has enough of this virus DNA inside of them to say they are infected. So even if you are asymptomatic, you still have the virus inside of you. So you will test positive. So that's not something you have to worry about. It's just that thinking about all this, tests are very time consuming to repeat. So each of these reactions is very sensitive, and there's a lot of different ways that it can actually go wrong. So looking at this image here to the left below, these are all different components that go into a single one of these reactions. So you have the DNA sample from the patient, the primers, which are the little Lego blocks of DNA that you're looking for, and then all the chemicals that you actually need to make the reaction happen. So when you think about this, how this would actually happen in a lab, it's very time consuming to repeat for thousands of times. And that's why there's a lot of innovation that's being attempted to go into improving testing capabilities, making the tests faster, making more tests happen at a time. And it's really an interesting problem. When you think about the flu, we understand the flu virus much better than we understand this virus. So there are more specific proteins or components of the virus that we can measure and decide if someone is actually infected. With this virus, because we really don't understand it as well as we understand things like the flu, and it's not as well characterized, we have to kind of go the long way. So until new testing methods are developed that aren't as intensive or as specific and as time consuming as this one, it's very difficult to diagnose everyone who shows some symptoms. Are there any questions on this before I go on?
Can you just talk about the 3 points at the bottom, the denaturalization, the annealing, what that means?
Sure. So DNA, if you look at this green image here, it exists typically in 2 strands. So it forms like a ladder where there are bridges in the middle. So the way this method works, the way that we try and fit the Lego pieces into the DNA, is you separate the 2 strands, and then on a single strand, you try and fit that Lego piece. So the process of separating them is called denaturation. That's that step 1.
Okay.
And it has to happen at 94 to 96 degrees Celsius. And that's very specific. It has to happen at that temperature. So once you separate that strand, in the second step, number 2, where that pink piece is going in, and that's the sequence that we're trying to match, what you do is you try and bind them. So that's called annealing, and that happens at 68 degrees Celsius. And then there is a chemical in this whole reaction that will then take that little pink piece and get the little piece, it will elongate it. So it will try and match the rest of the green strand by elongating the pink strand. And so basically what happens is you form a full set of double strands again, and that's called elongation. So then you continually repeat this process of ripping strands apart, trying to fit it, elongating it, and then ripping it apart again. And keeping that in mind, looking at how specific these temperatures are, that's where a lot of the reaction can go wrong. And you need very specific, expensive equipment to make that reaction happen. And that's called the thermal cycler, which you see in the bottom left-hand corner. And that's not necessarily something that a lot of places have access to, especially labs that aren't actually set up to do this technique. And that's one way that the companies who do this technique are trying to enter the market and develop new tests so that you can make that process a lot easier in matching the piece of the DNA to the entire strand that you isolate from a patient. Okay, so The next thing I wanted to talk about is social distancing in general. So it might be difficult to understand, especially when you listen to the news and you see that there are still so many people that are getting sick, even though we're implementing all these measures to stop contact with each other, and it can be kind of discouraging. So I just wanted to talk about the basis for this. And in order to do that, I want to introduce this R0 number, which is called the reproductive number. And what this describes is, on average, how many people will one person who's infected give the virus to? And the reason this number matters is because when we do projections and we try and estimate how long this virus will last, that is something we look at. In addition to doing projections and mathematical estimations, I want to show this graph of the number of cases to the fatality rate, especially looking at what happened in China. So China serves as a model for us to make decisions because they've already gone through everything. Now, when we look at the little circle, which represents the Wuhan province where the disease originated, we see over 4.42% fatality rate because they were very unprepared, and that's where the virus originated. So they really got hit before they had any idea how to mitigate it or really what it even was. But then when we look at the rest of China, where they had more of a warning, we see that 0.84% was the fatality rate. And what's important to note is that in the rest of China, social distancing was implemented a lot more effectively, where they had a notice and could start actually implementing these changes, rather than in Wuhan, where, like I said, they got hit by the entire virus before they even knew that it wasn't just a flu. So what this shows is that a lot more people are able to live when your healthcare system is not overwhelmed. So social distancing is the first step in making sure that we don't overwhelm our healthcare system. So this graph here is a mathematical projection, like I showed. So the numbers aren't exact here, but what the main takeaway is this red line with the dramatic spike shows the disease model and how many people will be infected if there's no social distancing at all. So you can see that it happens over a much shorter time span by looking at this x-axis where it says months since transmission established, or basically months since the disease started. And then you also see this green, the plot with the green line, which shows how many cases are reported when social distancing is in effect. So not only is the number of cases being reported shorter, but the thing to note is that the months since transmission established is longer. But overall, the fact that less people are getting infected, that makes it much more manageable within society in general. So that's really what's important to take away about the social distancing, is we want to avoid the high fatality rate, like was observed in Wuhan, where they really couldn't respond to the virus with social distancing matters, compared to the rest of China, where social distancing was very effective. Now, the other thing to consider is when you implement social distancing and then remove those regulations too soon, the blue line on the plot demonstrates what happens where before the virus is run its course, you start returning to normal life. And you see that the peak still happens much later, but really that's what we're trying to avoid right now. Although our numbers look good and we're seeing less and less cases every day, we don't want to initiate a resurgence of the virus by reducing the limitations on social distancing sooner than our healthcare system is really ready for. So with that in mind, there's a lot of factors that we don't really understand yet when we're deciding how long these shelter-in-place regulations should be effective. Originally, the R0 for coronavirus, the reproductive number, was around 2 to 3. So each person that was infected would infect 3 more people. But with social distancing, we've reduced that R0 to only 1, which is really powerful in terms of stopping the spread. But there's still a lot of things that we don't understand about the mechanism of the virus in general. For example, one thing that scientists are still looking at is if individuals that are infected will have short-term or long-term immunity. What this means is if you're infected with the virus, are you going to be immune from it for the rest of your life? Or are you going to be immune for maybe the next year? When we think about the flu, we get yearly flu shots because we only have short-term immunity. So we're not sure if that's going to be the case with coronavirus. And that's sometimes why you hear about this second wave that health professionals are trying to prevent that might happen if we only have short-term immunity, and we'll see a second outbreak in the winter season. In addition to that, social distancing really is important and changes the infection rate. So it's really hard to predict where the peak of our curve will be, or even what our curve will look like compared to these other 3 plots that I've shown here. So keeping in mind this plot, if we think about this in the context of Illinois, I know another question I've got a lot is, when will everything go back to normal? So really what professionals in the public health department and in other places are doing is trying to create these plots and basically predict the future of how the virus is going to spread and what social distancing matters are happening now. When do they think we can revoke the shelter-in-place order? And the criteria that will satisfy this is 14 consecutive days with a decrease in the number of new cases. So what this means practically is every day Illinois Public Health, the Illinois Public Health Department looks at this curve on the right of the number of new coronavirus cases, and they try and fit that to a mathematical curve. And that's why every day, the governor has press conferences and he says, we're looking at the numbers every day, and day by day, we're trying to understand when shelter in place can be lifted. So if you look at this curve of what things actually look like in Illinois now, you can see it's not as predictable and it's not as smooth as the kind of ideal curves you would get from a mathematical model. So with that being said, it's really hard to predict when will things go back to normal, or when will we hit the peak of the curve. Because if we look at the graph on the left, the longer social distancing goes on, less people are infected, but it happens over a longer duration of time than the peak happens without social distancing. Okay. Are there any questions here? All right.
No, thanks. This makes a lot more sense. What I'm trying to figure out is people protesting going back. And I think that some program like this would help them, I think, make a better decision.
Yeah.
Get into politics, but it helps me.
Yeah.
Thank you.
Okay. So Keeping that in mind, the thing that we've been talking about a lot, or we've heard about a lot in the news, is vaccines. And I want to mention why that's so important. The reason for that is because it kind of advances our immunity, and it gives us more capability to predict what happens with this curve, but also it'll stop people from getting infected, and it'll create herd immunity, which means that the people who are most vulnerable to this disease and may not be able to get a vaccine because they're immunocompromised are less likely to actually contract the virus because because no one around them is able of contracting the virus if they're all vaccinated. So I've gotten a lot of questions about why the vaccine development takes so long and how long it's going to be until we have one. So the easy answer to why it takes so long is just because there's really a lot of steps. To start with, you have to identify the chemical compound and the biological compound that's going to make an effective immune response so that the virus can no longer infect you. Now, these chemicals and these biologics are very complicated, and we're injecting them directly into our bloodstream, so it's very risky. With that being said, toxicology testing is very important. So this takes a long time in the lab. The compound has to be injected into an animal or injected onto cells, maybe in a plate. And you have to make sure that the vaccine itself doesn't actually cause any harm to the animal or any harm to the cells. So after that toxicology testing is done, you can submit to the FDA. And what that means is the FDA is actually going to review your formulation for any red flags of things that they might find toxic. And the FDA is going to tell you what kind of results and what kind of tests they want you to perform to show that A, your vaccine is safe, and B, that it's going to actually work in humans. This process the FDA is actually very, very thorough. And for that reason, this process can take a lot of time. For coronavirus vaccines right now, the FDA is doing a really good job basically fast tracking everything, but it's still very time consuming, and it's still a lot of work. So after the FDA approves your submission, a company can move into a phase 1 clinical trial. And in that clinical trial, the aim of the study is to look at the safety and any immune response to the vaccine that is unwanted. So they want to make sure that none of your white blood cells or any of your organs actually reject the vaccine or start attacking each other, and you don't have any autoimmune response where your immune system actually starts to attack you, because that's not something you want as a result of this vaccine. So after you complete your phase 1 clinical trials, you move into phase 2 and then phase 3. Where in phase 2 and 3, what you're looking at is not only is the vaccine still safe, because you have to maintain that, but now you can start to look at how well does this vaccine actually work at stopping someone from becoming infected with the virus. So one thing to keep in mind about clinical trials is it sounds pretty simple in terms of logistics when I describe it, but in actuality, each of these clinical trials has to recruit patients. So humans are actually the ones that this is being tested on, and you need volunteers to take this experimental chemical compound. And then there's a long time span to make sure that you don't see any adverse reactions. And the FDA has guidelines about this to make sure that it's safe. So it's a very tightly regulated market. So even after a vaccine isn't approved, something from a business perspective that people don't necessarily consider is the manufacturing of vaccines. A lot of times they contain whole viruses that have been attenuated or weakened or are actually dead. And sometimes it contains virus particles. So these virus particles in the dead virus actually have to be manufactured. I don't know if anyone's ever thought about how do you manufacture a cell before, but it's actually pretty complicated. And once a company establishes an initial protocol, even then you have to go through so many checks and balances that each one of these steps under normal circumstances can take about 6 months, which tends to add up quickly. And that's why another reason why pharmaceuticals in general tend to be so expensive is because the drug development process is extremely time consuming, because you want to ensure that the thing that you're giving people is safe. And just a lot of times it's a waiting game. You will inject an animal with a vaccine, and then you just have to wait, 8 weeks to make sure that nothing bad happens as a result of that injection. Another thing to consider is that sometimes vaccines for things like the coronavirus, companies will actually lose money on. So even though right now it seems like it's the most important thing, we don't know if there's short-term immunity or long-term immunity. So it could be possible that this is something that people need every year, but it's also possible that it's an injection that people will get once, and then it will, what they call stockpile, where the vaccine is never actually needed again for the initial round of vaccination. So this is something that companies have to think about and might actually hold them back from pursuing a vaccine for pandemics or epidemic situations. And even despite that, though, there are a lot of promising vaccine developments actually going on right now. So one of the biggest ones is by Johnson & Johnson. They are using whole virus vaccines, which means that it's basically just the entire virus particle, either weakened or dead. These types of vaccines where they use the entire virus particle tend to be a lot more extensive and a lot more complicated in terms of the chemistry that goes into the delivery. But Johnson & Johnson has historically gone after this approach, and so they're pretty knowledgeable in this entire process of vaccine development. Other types of vaccines are subunit vaccines, so where they might use parts of the DNA or parts of the virus within the vaccine instead of the whole intact virus. And then there's also nucleic acid vaccines where what this is using a specific strand of DNA or a specific strand of RNA to basically cause the virus to self-destruct by teaching your cells how to combat it. So in addition to viruses, there's also been a lot of different drug development programs that have been going on. Most of the drugs that are being investigated right now are previously approved. So vaccines are very, very specific to types of viruses, which is why it's very hard for us to leverage, for example, a flu vaccine with this current coronavirus. Drugs, on the other hand, are more easily repurposed. So what's happening right now is companies are taking drugs that have been approved for other uses by the FDA. And so what we can do is shortcut the safety and toxicology testing because they've already been approved. And we're testing that mechanism with the coronavirus. So a lot of the ways these drugs work, one of the biggest ways right now is by stopping virus reproduction. there are certain features that this virus has on its shell that are like spikes and are really important to basically latch on to the human cell. And there are ways that this is being targeted and stopped. Because these spikes and a lot of the other features of this virus, like I said, aren't specific to the coronavirus, this is much more manageable to repurpose than it is necessarily in a vaccine. So another method besides stopping the virus reproduction and inhibiting infection progression is to reduce the viral load within a person. And the viral load is the number of virus particles that are inside you. So this can be done by different excretion methods of the virus. But really the most common method right now is to stop virus reproduction. And there are drugs like chloroquine, which is in this chart here, that has been used for malaria, that targets coronavirus in the same way. So this is, like I said, a lot more promising right now than the vaccine development. In this chart, you can see 5 drugs already that are being tested and are promising for coronavirus. And the thing with the drugs is you don't have to have separate trials. So there are a lot of different methods for administration between vapor, inhalation, pills, intravenous infusion, that these can be administered, which also is really beneficial for this virus, where people are infected in their lungs but might not actually be able to inhale a drug all the way to the lungs, where it can be effective if they're having breathing issues or might be on a ventilator.
I have a question.
Yes.
So I hear a lot of like the blood drives that are going around for like the need for blood. Is that specifically for COVID patients? Is that supposed to reduce the infected cells and replenishing that with quote unquote healthy blood? Is that what that's doing?
I don't think that's what it is. I think that right now there's just a lot more sick people. And because of different complications like septic that might result from the disease, there's a need for more blood, but they're not necessarily using the blood as a treatment. It's just that it goes along with the overwhelmed healthcare system, where because you have more patients in beds, there are less beds available if you get in a car accident or have something else tragic happen to you that would require a hospital stay. So it's along the same lines with the blood donation, is there's a lot of bad side effects that could lead to organ failure is actually a common one with coronavirus. And so those are some types of things that you might need blood for. Okay. And just kind of as a result, there's reduced availability.
So I've got a question related to the previous one.
Mm-hmm.
What about where they're trying to get blood donations from people who have survived the virus, plasma donations? So they're hoping to get antibodies from those people and use them in treatment?
Yeah, that's a good question. So the antibodies are basically what goes into vaccines sometimes. That's one approach to vaccine development. Is the idea is to get your body to produce those antibodies. So it's not something, I think a lot of the things with antibodies right now is for research development, where they want to take someone's antibodies who's had the disease and look at the structure of them and learn about how that might be incorporated into a vaccine, or how they might be more able to encourage that antibody production through a vaccine. But right now, I don't think they have a concrete method for using the antibodies for treatment.
Thanks.
Mm-hmm. Okay. So I also wanted to talk about other types of technology that are being developed and have come out of the pandemic. The first is there's been a lot of ventilator innovation. So you've probably heard about ventilators because coronavirus attacks the lungs and reduces patients' capability of breathing independently. So what a ventilator does is it basically forces your lungs to expand and contract for you. Because when you have lung infections, the muscles that you use to breathe become much weaker and people are unable to breathe for themselves. So the ventilators force that air into the lungs and make the muscles work. So mechanical ventilators are a new type of ventilator that is being designed because it's easier to manufacture. Another type of ventilator accessory is an airway splitter. So if you look at this first image here, you'll see that they've attached that airway splitter, the attachment, onto devices. And now, instead of only being able to have one airflow supply per tube, you can now get two. So what that means is you basically double the usage of your ventilator from only being able to be used on one patient at a time to two just by splitting the airflow. There's also been some mouthpiece developments to try and stop to try and make it more sanitary for healthcare professionals. With that in mind, thinking about the healthcare professionals, we all have heard about the shortage of masks. One type of mask that is currently being developed is self-sanitizing. And the way this works is there are particles within the mask that are aimed at actually killing the virus upon contact. So as the air flows through the mask or across the mask, the virus is deactivated. And that actually enhances the lifespan of the mask and lets healthcare professionals use it longer and more effectively without necessarily having to clean it or throw it away right away. 3D printing is also something that has been widely used for masks. The reason that is because there's a lot of 3D printers that aren't at manufacturing sites and can be used in academic facilities. Face shields, like in the lower right corner here, have been one thing that people are 3D printing a lot. One thing that's really convenient about 3D printing is you can pretty easily change the size and the metrics of the things that you're building. So in the top right corner, one group of researchers has been building customizable 3D printed masks. And what this means is that each healthcare professional, each nurse or each doctor will have a mask that conforms to their face, which makes them safer because they have less of a risk of having airborne particles go through it if it's too large or if it's too small. So that's really beneficial, and it's reusable because it's 3D printed. So the only replaceable component is a filter that goes in between the face part and the outside part that actually comes in contact with infected patients. So that's really beneficial in filtering out the air particles, and it's potentially less wasteful because it traps the particles. So this is some really cool discoveries that researchers are making and coming together. The face shields are also really good because it protects people's full face at once rather than having a lot of separate components. Because we don't know a lot about the virus, there's a lot of work that's being put into how the virus might be transmitted through your eyes. I think recently the CDC put out an announcement saying avoid wearing contact lenses because you want to avoid touching your eyes and touching your face. Mm-hmm. That's why the face shield is really effective, and just like the 3D printed mask, it's customizable if you're 3D printing it. So you can make sure that it fits securely.
Quick question on the splitters.
Mm-hmm.
Wouldn't that be, would there be cross contamination if you're splitting the airflow and you're going to two different patients?
So the airflow is coming out of the machine clean. So within the machine, there are like sanitizing processes. A lot of the germs also get stuck inside the patients, like they have tubes put in. So those tubes have to be cleaned regularly. But they're mostly kept separate. So there's not too much contamination. But that's a really good question.
And I assume if the airflow is split, that the source airflow is high enough pressure that splitting it maintains the pressure that you need.
Yeah, you have to definitely change the parameters that from one patient to two, but it's still effective for most ventilators.
Thank you.
Mm-hmm. Okay. So one of the last things I wanted to talk about is managing stress. With all this information, it can be kind of overwhelming in terms of, especially when you start to think about the things we don't know, and we don't know when the shelter in place is going to end, and there's really no way to predict it right now. One of the biggest things that people have said is very helpful is to take breaks from the news and the negative information every day. I know it's really important to stay informed, but also it can be kind of depressing, and that's something that researchers have found is people need to take breaks from the news. So try and maybe schedule like an hour or two a day where you just disconnect from everything. Also, it's important to keep your body healthy and try to be going on walks. With the nice weather we've been having, it's been pretty easy to take advantage of this, but you don't want to get too cooped up in your home. It's nice living in the suburbs instead of Chicago right now, I think, because all the parks are closed down there, but here it's easier to maintain social distancing while still being outside. In addition to maybe planning breaks from the news, it's also important to plan time for leisurely events and things that you enjoy, and try and do that every day. And then the last thing is to check in on your friends and family and try and stay in constant communication, even if you can't see them face to face. So I also wanted to talk about the best sources for information. Number one is medical professionals, people like doctors who can diagnose patients. I'm not a medical doctor, but so they know more about things like your symptoms or when to see a doctor or when you need to get tested. And I know a lot of places are having like call lines that you can reach out to if you have any questions regarding that. Another thing, if you are interested in anything I talked about, like the ventilator development, drug or vaccine development, even how the virus is spreading and the mathematical projections I went through, there's a lot of research journals that are open source. And these journals are the places where scientists write up reports on everything they've done. It's really quite detailed, and you can read about the different problems and where they got their ideas from and how they're implementing this in the clinic. You guys had a lot of really good questions about antibodies and things like that. So that's all types of information that if you're interested in, you can read more in the open source journals. Also, the company websites are very helpful and typically accurate in the type of research that they have going on. I know for me, I didn't used to think of companies as places that performed research until I started working at places like Johnson & Johnson. I've put their link here because their website is very manageable, and they do a really good job of updating everything that they have going on. So if you go to their website right now, one of the things you can read about is the path to the coronavirus vaccine, and it's really interesting. The last one that's on here is the CDC website. The CDC has been very on top of answering all the frequently asked questions. If you were interested in the t-shirt face covering diagram that I had, that's where I got that resource from on how to make it and the types of requirements that your mask should satisfy. They also have guidelines for sewing masks and the dimensions that the mask should have to fit on your face. So that's a really good resource to use as well. So I'll take any more questions. Thank you guys for listening. And I've put my email here. If you have any other questions about the open source journals or how to access them or anything else I talked about today, please feel free to shoot me an email and we can talk about anything. So thank you guys. And does anyone have any more questions?
I had one question on the when they're doing the test with that Lego piece.
Yeah.
So does the test have to meet a certain threshold? Because when you're stripping the DNA strand, it's a long strand. So I'm assuming that it's going to fit at some point during the DNA strand. So are you looking for a certain threshold in which it matches the original DNA strand? Does that make sense?
Yes, that's a very accurate question. So I didn't talk too much about the actual chemicals, but what happens during this reaction is every time the Lego piece binds, it's attached to a fluorescent molecule. So the machine, the thermocycler, measures the fluorescence of the solution. So the more Lego pieces that bind, the higher the fluorescence is, and there's a certain fluorescence threshold. So at a certain threshold of fluorescence, they can say that there's a significant amount of the DNA. So with this method, it's really easy, not really easy, it's more likely if the sample is collected incorrectly to get a false negative, because there's so many things that could go wrong. They could say, oh, you didn't meet the threshold, you're not infected. It's more common to have a false negative than it is to have a false positive. You're really not going to have a false positive with this method. But there are some risks where if the DNA is damaged or the sample isn't collected correctly, that Lego piece doesn't bind in the right way, and then you don't get the fluorescent threshold that you need.
Thank you.
Any other questions? I'll go back to the slide with my email. So again, feel free to reach out to me if you do think of any questions. I'd be happy to answer them or direct you to the resources that I used to make these slides.
Yep. And then we'll also put it up on our, like I said, on the library webpage and our social media channels as well.
All right. Thank you, guys.
Thank you so much.
Thank you.
Thank you.
Thank you.